Welcome to the 47th session of day 6 of WADLA 3.0 on topics recent trends in deep learning and application. So today we have with us Dr. Ramanarayan Mohanty from Intel Lab. So he is going to talk about the deep learning application in biological system. So before his talk, so I would like to introduce him. So Dr. Ramanarayan Mohanty is a research scientist at Intel Labs, India. His work primarily revolves around the graph-based machine learning and deep learning research in the domain of general science. He is a graduate of the Indian Institute of Technology, Kharagpur. He earned his PhD in the specialized field of graph-based machine learning for hyperspectral imaging. His scholarly contributions have been recognized and published in numerous reputed journals and conferences such as IEEE, Elsewhere, and Wiley. Dr. Mohanty's core research pursuits lie at the intersection of graph AI, machine learning, deep learning, and computer vision. Through his robust research and where he continued to make significant strides in this rapidly advanced field. So welcome, Dr. Mohanty. So now session is over to you.
Thank you so much, Dr. Rakesh. Am I audible to everybody? Hello.
Yes, you are audible.
Okay, thank you so much. And also, I want to. This is my honor to present on this particular workshop on deep learning applications. So, okay, so today we'll basically talk regarding the deep learning applications in biological systems and how it is revolutionizing the drug discovery, protein synthesis, and in other healthcare sectors. So, yeah. So let's start. And the topics I am basically going to cover today. Yeah. Okay. So the contents mostly revolves around like the introduction part. I will just give brief introduction to biological systems and some overview of biological systems and the challenges. What are the challenges basically we faced while we go for any kind of means while approach any biological systems, and then the deep learning fundamentals, some basic fundamentals of deep learning, and and then the deep learning applications in drug discovery, how it is applied in drug discovery case, and then the applications in protein synthesis. And then after we'll discuss something regarding beyond drug discovery and protein synthesis, because these two are very hot topic recently. And beyond the drug discovery and protein synthesis, what are the other topics we could do research and we could look into, and then some conclusion and future scope and question answering. So excuse me. So while I was just skimming through the 2023 ICLR papers, and means if you look into the major topics what are covered, so there are definitely large language model is the hottest topic currently it is going on. But apart from that, what are the trends actually approaching, and what are the trends that we look forward for the research, and we could there are a lot of scope in doing the research on those things. And if you look into those things, I mean in the top 10 list, that graph representation learning and molecular modeling and geometric deep learning, these basically these topics are gradually increasing day by day, and the demand for these things. And since computer vision and NLP, those things are quite popular, but these particular research areas are gradually and also creating a lot of buzz in the industry as well as in the if you look into the pharma industry or any healthcare sector, everybody is going for this artificial intelligence, means integrating artificial intelligence into the system. And so that's why while we go for the structured data like those sequences or the images, so those out of those things, if we come out of this particular structured data, if you go into any unstructured data, the AGI type of artificial intelligence comes up, like artificial general intelligence. So how to deal with these things, and those things are basically targeting the normal general science area. The research is happening, what are means whatever we have done in chemistry, physics, biology, those things are very traditional way till date. But now how we will do the research in using the deep learning there. So this is very trending and very hot, also some of the hot topics in the industry right now it is coming up. So let's come into the different applications. If we look into, these are very, means I have just abstracted out, so very mm, some uh, some application areas, those are quite popular, and there are a lot of work is happening in this particular application areas, like facial recognition, speech recognition, image recognition, and natural language processing, recommendation system. And one of them is the healthcare sector, and healthcare sector is actually booming right now. It is it is in the initial phase of growth, and there are a lot of work is happening, and a lot of people are going into this particular field targeting. And means I won't be surprised if after 3 4 years we will see something, you don't need to go to any doctor or something, everything will be done, and based on your own, means you won't get any generic medicine, you will get any specialized medicine for yourself based on your gene and based on your medical conditions, earlier medical conditions and all. So this kind of research are going, is actually happening, and people are working a lot in this particular type of thing. So once going into the biological systems and biological deep learning application of the healthcare sector and all, so we need to know what are the basic challenges we face while we deep in, means go into these particular biological systems. so these are some challenges that I have abstracted it out. There are a lot of things are there, but still I try to some major challenges that we face. Scale up complexity. So the biological data is so huge that it's actually starts from genetic level to a complete ecosystem. Means from genes make some molecular networks, then from molecular networks to it becomes cellular networks to organ networks, then organ networks to individual, then individual to network of individuals. So think that way, means how complex it is. And if we for a means for a single human being, the number of genes, if you see, there are 6.4 billion letters are there, that is a real human genome. And for a mitochondrial it is 3.2 billion. Think about how many species, how many individuals are there, how many different type of organisms are there. So this is very scalable system, and the data is very huge, and to deal with those particular things we need a complete infrastructure. So, and another thing is the data overload. So what happens, since the system is so complex, the network is so complex, so the data is also is getting overloaded. So each means, just think, for a single person 6.4 billion, then another person another 6.4 billion. Although there are a lot of similarities there, but still there are some new genes are there, because all persons are not same. So think that way, how many genes or how many data is coming up from these particular systems of challenges, means biological data. Then interconnectivity. These biological systems are very neatly interconnected between each other. Means as I told, like genes are connected to the multiple genes. Then if we come below the genes, we will come to protein level, then genes, from genes to different sequences, amino acid sequences, from amino acid sequences to proteins. So there are lot of interconnectivities there. It is not very straightforward things, and it is means, if we look into the connection between, or the complexity between the genes and the genes to molecular networks, it is too complex, and they're very interconnected with each other. So that is another level of challenge. Then another one is nonlinearity. So it is not that simple that we make some input in one condition and we'll get proportional output mm, in the other end. Uh, it won't give that way. It gives, there are a lot of things getting happened, so it is not linearly changes. There are a lot of nonlinearity are there in the data. Then genetic complexity that I already told, and apart from that, the interdisciplinary nature. So while we look into biological system, it is not purely biology. We look into a lot of other things, like it is also chemistry. While we go into the protein level thing, we have to look into different molecules. So chemistry is there, then physics also there, then computational, since we are looking going through computational analysis and AI based analysis. So then in that case, computer science also involved there. So a lot of interdisciplinary nature means interdisciplinary things are involved there. Then one of the major challenges is the ethical consideration. It is we cannot use directly from anybody's gene. There are a lot of ethical constraints are there. Like there are a lot of I will say there's some regulations are there. Some we cannot apply the some drug or something on any animal. It cannot be done. We cannot test it on. It means we can use any animal as a testing test bench or something like that way. We cannot do that way. Okay, so there are a lot of ethical considerations are involved. Then computational limitations. So as you see the scalability of the data is generating every day, and so the computational power whatever we have is not sufficient to do the processing all those data. If we go through the traditional way or the uh, normal means the way whatever previously people are doing the research on the biological science, so we need something different that can be done, that can be do some faster processing as well as do things very fast and uh, get some proper info means proper data, or the we could infer something from the data very robustly. So that's why the computational limitation is nowadays a huge thing. Then it's the redundancy and robustness. The human body or the biological things are very robust. We adapt to different type of situation, different type of condition. So what happens, so just to keep the robustness, if means I will give one example. Like a person, means if we consider a person, it has 6.4 billion human genome, means real human being has a 6.4 billion genomes, or the basically the letters ATCG, those things are there. So if I go to another person, it also has same 6.4 billion, but if we see those things, they have same, but the two different person have same structure, body structure. But so in that case what happens, lot of genes are common there, but there are some dissimilarities also there. So there are some new genes are coming up. So that's how the redundancy is also there. Means lot of, that means when the data is coming up, if we collect data from multiple people, the gene information, then we'll get a lot of redundant gene there, and those data are very huge. So in from there we have to filter it out the uh, discriminative part or the most important part for the particular person. So that's how the redundancy, and that is also one kind of robustness. Like since that this human body is more adaptive and also there way there are a lot of uh, genes are there which are actually more robust. So those things are actually some kind of challenges that we face, and also lack of standardization. Till now it is in the nascent stage, it is basically in the infant stage, if we say. So people are just starting research in this particular field, and they are trying to do some groundbreaking work. If you know the AlphaFold 2 and all those things, basically some groundbreaking research they have done. And so but still now there is no standardization is there, there is no government what so means everywhere there are some standardization. So the standardization process is, means it is in the starting phase. It hasn't completely everything, every data is not completely standardized, because we haven't collected all data. We are just right now we are collecting the data. So this is another level of challenges. So these are the major challenges I described, and we'll see those things in the latter phase. So so here I'm basically focusing on two major cases: the drug discovery part and protein synthesis part. So if those are some broad challenges we see in the biological system, but if we come into drug discovery and protein synthesis, so there are some different kind of challenges in each part. So what happens in drug discovery? The if you see the normal time trend for drug discovery is 10 to 12 years for a single drug, and that cost billions of dollars for creating a single drug. That will discuss in the latter phase how it is going through. Then there is a lot of chance, means 90% chance is that that might fail after 5 or 10 years while we coming to the testing phase. Then as I told, there are a complex biological systems, means lot of means one single drug, we are taking a drug for fever, but we don't know that drug is either effective for other organs or not. We are only concentrating, okay, we are taking that drug that is only curing the fever, but if that will affect something adversely for some other other organs or something, then that we have, that complexity is there. Then personalized medicine, personal means we have to look into, everybody's gene is not same. Some genes are same, but some discriminatory parts are there. So one drug is effective to me and that might not that effective to some other person. So that's how the personalized medicine is coming up, that gene factor is coming up. And antibiotic resistance, everybody's body, means every person's or every animals has some bacterial things are there in the body. So what happens, they create some resistance to the outer any kind of substances coming into the body. So those things they have to look into, and they have to uh, analyze those things so that their drug should be effective to the to the disease as well as to the person. Similarly, if we look into the protein synthesis part, so prediction of protein structure, protein structure is one of the most complex structure that we ever see, and predicting that protein structure is one of the major challenge till date. Till 2020 we are basically facing, but after 2020 during the CASP challenge, DeepMind has basically that introduced the AlphaFold, and that did a tremendous job, and that is one of the best research in this particular decade that we have seen. And then synthesis of large proteins, it is not all about predicting the proteins. Sometimes we have to synthesize the proteins by ourselves so that it could be more effective to certain diseases and all. So those things we have to do, we, because a lot of compounds are there, we don't know how it is, and we have to figure it out how to synthesize the proteins and all. Then post translational modifications, like after synthesis, after synthesize the proteins, the proteins go through some kind of uh, means modifications, in the sense they are callings, like some glycosamine kind of some enzymes or something comes up, and that it changes the structure of the protein, and it again folds into different new structure. So that the folding of the protein also getting changed. Once the folding of the protein getting changed, then it might affect into different sometimes it acts positively, sometimes it acts negatively. So those things we have to look into. Then protein folding, and yeah, protein folding means how the protein is getting folded, that we have to figure it out. That basically the AlphaFold has done this particular protein folding work. They have used the AlphaFold architecture to determine the protein folding. And then doing those things means how efficiently we are doing it, and how fast we are doing it. Those are another level of challenge that's mostly coming into the high performance computing type of systems and all. So these are major challenges in drug discovery and protein synthesis. So there are some required solutions to overcome these challenges, like some efficiency, I mean, how efficiently we could do, how precisely we can basically look into those particular proteins or molecules and all. So these are basically some solutions that overcome the challenges. Efficiency, precision, some scalability issue, how to deal with the scalability issue, and personalized medicines that I told, and how effectively we predict the protein structure and all those things. Also predictive power also one of the things. Then handling complexity, data management, sustainability. These are basically some overall solutions that we have to incorporate while we are going to solve these kind of particular problems. So yeah, these are some basic fundamentals. I won't go in depth of these particular things. These are everybody's know some basic building blocks for deep learning architectures and all. So and yeah, these are some flavors of neural network architectures that basically utilized in some these biological systems. These are some traditional. Basically, I won't say these are completely new. These are some traditional things like CNN, DNN, LSTM, then autoencoders. Then actually, this graph-based networks actually make a lot of impact in biological system because the molecules, the networks, everything are inherently represented as graphs. So graph-based network like GNNs and graph neural networks, graph convolution networks, MPNN, and these things are message passing neural network. These are actually making a lot of impact. And also, what nowadays people are doing, people are mostly collaborating, combining this GNNs along with the transformer. So that gives some graph transformer kind of things. Those are quite uh, promising field right now. People are working, and there are a lot of papers. If you see the ISLR and NeurIPS papers, you will get a lot of papers on this particular domain. So these are basic architectures we utilize in the graph based, I mean the solving these particular biological systems and all. So yeah, this is quite common, some laws and optimization techniques and all. So basically those different laws, functions, means optimization like stochastic gradient methods or momentum, ada grad, ada delta, those things basically utilized in this particular thing. So I'm not going into on this the common frameworks we used. And majorly if you see the PyG and DGL, these two are majorly some graph based libraries, and those graph based libraries are yeah, the PyG is a Stanford based one. This one is I think Amazon has developed this particular DGL library, and those libraries have, if you look into those libraries, they have given a lot of examples there. So those examples might, that means for some majority of the examples are coming for the means are basically for this particular biological systems and chemistry and all. So if you could, you will get good idea about why this graph is more towards these biological systems. So going to the building blocks for deep learning in biology. So if you look into the building blocks, so these are basically divided into 5 blocks. So one is this data collection part, the architecture, deep learning architecture. What are the application we are targeting, and the hardwares as well as the tools or frameworks we basically use for this particular type of system. Yeah. So the data collection, if for biological systems, we collect data through different channels. So we could easily say these are basically multimodal data. We cannot concentrate on single sequential data, nor image data, nor means one dimensional speech type of thing. So there are multiple domains are involved here, like ECG, ENG, those things, and electromyogram, cardiogram, those things basically used collect data from the heart rate and all. So yeah, these are basic, the all the channels and all the kind of data collection instruments and sources. Basically this type of data, some images are also involved, some DNA sequences and involved. And if you see the tabular data, like whenever we go to the doctor gives the prescription. So from the prescription we get all that electronic health record, basically we call those things. So those data we collect, and then we use some deep learning. So this is the complete architecture. And yeah, for these things the majorly we look into some kind of hardware stuff, because everything cannot be done with, to be specific, there are some problems are there which are very good at CPU based systems. Some are there which are very good at GPU based systems. But there are some problems, like I will say, like while going for some genetic analysis, there are different stages are there. Some stages normal CPU, GPU, those things won't affect. At that particular time, they look for some kind of specialized ASIC type of thing. So that's why the broad spectrum is there. Means it is not that easy that okay, we took the data, we did it the processing, put it in a CPU. Because if you go for determining that particular disease, think about COVID case. If you won't get the drug uh, prepared within 1 year, then what would be the situation? So that's how means. And yeah, AI also played a vital role there, that we discuss. So yeah, so today we'll basically discuss major two things: drug discovery and repurposing, and protein structure prediction. There are some other applications also there, means medical imaging, genomics, personalized medicines, disease diagnosis, prediction, electronic health record. So these all are means emerging research areas. But yeah, today we'll just focus on these two things. So let's go into the drug discovery part. So as I said, the drug discovery part, it's basically takes a 10 to means 12 to 15 years or 16 years of time frame, and traditional way how it is done, and it costs millions to billions of dollars for a single drug. And that basically covers three phases: one is the discovery phase, one is the development, and one is the clinical studies. So discovery phase, basically they have the objective there, what is the disease, and they there is a research team is formed, and they are try to localize the molecules or synthesize the chemicals that required for the drug. And you won't believe the in the number of molecules are there, like not billions, it is in exascale. I'll come to that part, how big it is, and that is one of the major challenge. So novel means that part, in the discovery part, basically they try to figure it out or to shortlist the molecules. Then they go to the chemical testing, as well in the test tube manner, or in the lab rat or something, some animals, they test it on animals and all. Then after, they go for some formulations of this particular drug, and then they go for some files and there's some administrative procedure. And after, once the application is new drug application is filed, then they go for testing phase. So in the testing phase, they basically go into three stages. The phase one basically look into they test it on normal healthy human beings, so that it could be they could understand the toleration of the drug. And then after, they go for some few patients for that particular disease. And then they have, after that, they will go for large scale from different ethnicity, different, because they want to make the drug generic for different means continent, different, all those. They look into all the diversity, and they try to get the patients from different diversity and test it on them. So they want to check how it is behaving on different diversity, because we know different diversity are different means geological conditions, and as well as different ethnicity. So people's gene differs. So at that particular point, how it is behaving on individual people. So that's why they try to go this extensive testing on the drug. Then after, they file it, and after that, it got basically approved for the marketing, and then after, we'll get the drug in the market. So this complete flow takes 12 to 16 years, and it costs billions of dollars. So if think about, in during COVID, if it will go through this procedure, what will be the condition right now? Just imagine that way. So there comes how we could all these different parts, how we could do those things or simulate those things or do it computationally, so that we could make it more faster way. Because those things are very traditional way takes a lot of time, because they have to take each and every molecule, they have to test, and that testing, they will means basically filtering the molecule from a large, you can see if there are 10,000 compounds, they have to get one compound, that compound will produce the drug. And getting that is a huge process. That such process, if we do computationally or we use any AI based techniques, that would reduce our work from years of work to some days of work, kind of that way we can do. So this is how the DL method is basically integrated with the drug discovery process, like design and discovery phase. Like means what are the drug target things we are looking for, or disease we are looking for, how we will do means how we will get the molecules or the synthesize the atoms or molecules for this particular disease. So those things basically design and discovery phase. So here basically synthesis prediction of de novo drug design means, and then the molecular property prediction. So here these things, if you go for the traditional way, it will take a lot of time. But if we go with the DLMS method, like some generative modeling or reinforcement learning, if you use this kind of techniques there, then it could shorten the time frame, and it could give us very accurate and more in a very less time frame, and that actually matters while it comes to the any kind of pandemic situation or something like that way. So then it comes preclinical research. So virtual drug screening and target detection. Yeah, that is also on that part, means that screening from 10,000 compounds. So if we screen it to 50 compounds, from 250, we'll screen again 5. So narrowing down to single compound is a big challenge. And it takes a lot of time, a lot of effort, and a lot of money. So how to reduce those things? So that's where we could introduce the deep learning models, and that can be done through some kind of generative models as well as reinforcement learning. So some new techniques, some representation learnings are also there. They can use those things. Then while it comes to the clinical research part, clinical trial treatment means mostly what happens, if you see, if you have any pandemic, you don't have that drug is currently available, you have to get the drug as soon as possible and do the means, because if you won't get the drug, if you don't have any this is actually do or die condition at that particular point. So that's why what happens, the speed needed at means very importantly, means it is very necessity there. The speed means the time to production is very essential at that particular point. So then this clinical trial recruitment and drug repurposing, those things, basically drug repurposing means some drug is there, means if you think like chloroquine tablet, that during COVID, the people are using chloroquine tablet because there are some compounds are there in chloroquine that were quite effective against the COVID. Since the chloroquine is a proven and available drug, people use those things. So that is basically called drug repurposing. So how could they figure it out? If they try to figure it out through traditional way, that will take time. So they use some deep representation learning methods or graph embedding methods. From there, they try to figure out the molecular structure of the graph, and then they try to find the molecular structure of the virus and all. They try to fit, okay, it is fitting properly, then okay, this is effective. So from this way, they basically try to um, uh, shortlist this particular or the repurpose the drug. Then similarly, automatic drug means some documents reviews, means whatever the FDA reviews, those things can be done through some predictive models and all. And some adverse drug effects, polypharmacy or side effects. Polypharmacy in the sense, what happens while we are taking medicines, while we go to doctor doesn't give us a single medicine, they give us two to three kind of medicines. They give for each kind of symptom or something, they give one medicine, but doctor doesn't know what will happen if you take two to three medicine at a time into our body, and if they will chemically react with each other, what will be the product of that reaction and output of that reaction, and how it will affect, whether it will positively affect or adversely affect to the body. That is called polypharmacy. That means multiple drugs we are taking. So those things can be detected, those things can be done, and those research is going on, and that basically graph based machine learning or deep learning techniques are used there for detecting this polypharmacy effect and all. And then food drug interactions. So how to determine drug, rather than taking drug from the food itself, there is some drug, uh, uh, some kind of basically I could say drug like some molecules or some compounds are there in the food that is effective rather than taking the drug. So those things are there, those things we can determine through some deep representation learning and graph based embedding. So yeah, so basically that virtual screening part. So as I said, so one approved means recent times, the one approved drug average cost is $2.6 billion. But what happens, the number of atoms, means the number of drug-like molecules in the universe is comparable to the number of atoms in the universe. Think how big it is. Means even it is, they are calling it in exascale, that is 10 to the power 10 to the power 18 number of molecules are there. Think 10 to the power 18 number of molecules. From there we have to determine one single molecules. So if we go with traditional method, then it will take 1000 billion CPU years to do these things, physics based traditional method to simulate those things. So there it comes how to use deep learning, make it much faster and do those things. So Mila, basically they have done some kind of work like they are calling lambda zero. So they are like Monte Carlo tree based search. They are defining that search, and that search is mostly um, used to determine the components or drug-like molecules from the universe. Actually, we are collaborating with Mila for this particular project, and we are working on this. But this project is a very huge project, and work is going on. So, how to determine a single drug-like molecule from this big universe of molecules, and how to make the search that so fast so that it could be visible, means it could be done within certain time frame. So this is one kind of example I told. uh, then different then target identification. So how this the particular drug is effective to the particular targeted disease and all. So for that thing there are different kind of data is necessary. So the data is basically what happens, we integrate like some genetic information from the disease, virus or bacteria, and other some disease patients. So the genetic information plays a vital role in detecting that particular molecule. So from the genetic information from the gene, we get the protein structure. From the protein structure, we determine here how the molecule structure is there, how effectively it is fitting to the protein structure, and that could be effective towards curing the disease. Then other things like pattern recognition, predictive models, generative data analysis, identification of protein structures, means yeah, those things whatever I told. And then network analysis, how this particular biological network is connected or something within that particular disease person or something. That way the network analysis, single cell analysis, or drug repurposing, these particular kind of struggle means, but these things are involved in determining the drug target identification, how to identify the target or the uh, I will say like how to determine that particular molecule that will be effective towards the particular disease. So that's how we do the drug target identification. So here basically deep learning in accelerating the drug discovery. So what happens, so while we go for any kind of right now, as I said, it is taking a lot of time. So we have to put deep learning in different stages of the drug discovery. And one of these things is, as I told in the first thing I told, searching the molecules, that we are using some lambda zero or something, that kind of. Then target detection part, we are determining, means we are using different components and we are using some kind of different techniques there. But while coming for the complete deep learning means drug discovery process, to accelerate the process, so we basically call it as in silico medicines, or in silicon, in the sense we will do it in the computer. So everything will be done from target detection till the medicine. Everything can be done in different stages and in the computer simulation manner, or through any AI techniques. So that's how the different means whatever was taking time, those things can be reduced to a lot if we go with any kind of computational method or some kind of AI based method, like pre-training, some fine-tuning those particular molecules we got. Then we go for testing those models on this particular, by considering some data from the input, means the drug molecule, and as well as data from the disease patients or disease virus gene. So then we go for some predictive analysis and all. So those things basically involves a lot of different stages are there. So if you see this graph neural network in drug design, so graph neural network is one of the major uh, deep learning architecture used in the design and drug discovery part. So the majorly it is used in this molecular property prediction, this high throughput screening of the molecules. That whatever I told, that lambda zero, they use graph based techniques there for the high throughput screening of the molecule. Then de novo drug designs, then protein engineering, drug repurposing, everything use the graph neural networks, like some variants of the graph neural. It is not purely the GNNs only. Also some variants of the graph neural networks, and sometimes it is also, while the data is very huge and it needs more parallelized techniques, they are the sometimes we use this like architecture, means I will say transformer plus means GNN based architecture, graph transformers kind of thing. So this is and also MIT and the collaborators, basically they use a GNN model called chemprop to find a molecule called halicin that is basically used as means exhibit some antibiotic properties against some bacteria called E. coli, and that they published in 2020, and that is one of the groundbreaking work they have done for the means using the deep learning means applications area. So yeah, some case studies like there are some in silico medicine and drug design, means basically they used some GANs and reinforcement learning to design a novel drug in 46 days. Think 10, 12 to 16 years to 46 days, how effective it would be and how fast it would be. And that they have done. Similarly, some like benevolent AI and drug repurposing, they used some kind of drug that is basically a rheumatoid arthritis drug that they used for repurposing for COVID-19 treatment, and that was also accepted by National Institute of Health, and in that treatment guideline they have mentioned that one. So these things were done through some deep learning techniques. And some recursion based, means there are a lot of pharmaceutical things and the deep genomics and deep and genetic medicine. They also announced some AI platform that discovered novel treatment for a new disease called Wilson disease, and some rare genetic disorders. So those things majorly, if you see the genetic disorders and all those things, majorly nowadays are the drugs and the treatment procedure, everything they analyze the gene component and they use some kind of AI techniques there and going for the treatment of those diseases. Then this atomize and drug discovery, they have actually developed a um, AI technology they are calling AtomNet, and that AtomNet is used to basically discover a drug candidate for multiple diseases, and also that include Ebola and some multiple type of cancers. So they used that AtomNet architecture for determining this drug, means yeah. So these are basically some success stories of deep learning driven drug discovery. So then we'll go into the deep learning applications in protein synthesis. So if we look into the challenges of protein synthesis structure, I have already told, like majorly the scalability and that's I won't waste time here. This I have already discussed these things. So just for your information, like one protein, one protein is consist of several amino acids, and there are 20 types of different types of amino acids are there, and those particular sequence of amino acids basically create a protein structure that we'll discuss here. So if we look into protein folding, the protein folding is majorly how the protein structure is there. It is very complex structure, and a newly synthesized protein has, there are four structures are there. One is primary, secondary, tertiary, and quaternary. The primary structure is nothing but, if you see, this is the primary structure, and these are the sequence of amino acids there, and those amino acids basically form the primary structure. Then once the folding begins, the sequence basically determine how it will fold. the sequence of amino acids basically determine how it will fold into that particular structure. So the protein folding means then in the secondary structure you will see this is the secondary structure, and in the secondary structure you see something called alpha helix and beta sheets. And then once these two are again folded, these two are folded to give the tertiary structure, and this tertiary structure is the 3D alignment of the proteins. And then after that, once the 3D alignment, three-dimensional structure is formed, then again those three-dimensional structure is again folded back to give a quaternary structure, and that quaternary structure is the most complex thing, and it is the protein molecule. They are calling it protein molecule. And based on these structures or the folding of the protein, they determine how it will be docked with other protein molecules, or how it will bind with other protein molecules or disease molecules or genetic genes. Or that's how the genetic behavior determines. So based on the protein structure or the folding of the protein, the genetic behavior determines. If we know how it will fold, then we could able to know how it will fit into other disease molecules or other uh, I will say what I will say means, yeah, but disease as well as some other protein bindings or something, so that it could be properly fit into those things. So this is how the protein folding happens. So then the deep learning approaches for protein structure prediction. So the protein structure prediction has multiple deep learning methods are involved, like predicting protein folding, like CNNs and GCNs are mostly used for predicting the protein folding structure. So AlphaFold itself has some transformer-based model. So they have used transformer, also some graph-based networks there. Then a contact map means this particular is using some DCNN kind of new architecture for this particular determining this contact map prediction. So means if we predict this particular thing, means it means that by learning from known protein structure, the model can accurately predict the residue contacts. So means that at the protein structure determination, how the structure that can be determined through the using the contact map prediction. Then predicting protein flexibility, how flexible it is, how it would be fold or how it would be changed with respect to different that chemical interaction and all. So those things can be also predicted using some deep neural networks. And then protein interaction prediction, how each protein is interacted with other proteins, those things can be predicted using some graph neural networks. And then also the predicting the secondary structure means those alpha helix beta sheets, those can be predicted using some kind of reinforcement recurrent neural networks and all. So there are there are a lot of other things are also there. I mean, predicting there are some the protein foldings are there. There's some subsequences are there also. Those things also we try to predict. So there also use some kind of networks like diffusion based models and all. So though there are a lot of things are involved there. So I am just going through some very abstract way, because itself is single topic itself is one huge research area. Then protein folding and protein interaction prediction using deep learning methods. So basically the protein folding and protein this particular protein prediction method is the AlphaFold 2 and all those things majorly used for protein folding prediction method. And some protein interaction methods, there are some deep complex network and deep interface network also they used to determine how one protein is interacting with other protein and all. So those things are utilized. And these deep complex networks majorly they use GNN based methods. And also in AlphaFold you will find majorly it is transformer and also reinforcement learning. So this is not purely transformer based methods. There are a lot of reinforcement learning is used here. So the case study means, this is the AlphaFold architecture majorly we are looking into. So the AlphaFold architecture they have they are considered, they are taking the input sequence, then they are going some genetic database search to determine that, and this multi sequence analysis part, they are determining those particular sequences. And majorly, if you see, the deep learning part is here. Evo is nothing but a transformer model, and that transformer model has, I think, 48 layers, means 12 layers with 4 blocks, means the parameter sharing is happening over 12 layers in each block. Within a block, the parameter sharing is happening, so they have turned it into 4 blocks, and then there are 12 layers. And yeah, so there are total 48 layers are there in this particular Evo firmware. Yeah, 48 blocks. Then there's some structure modules are also there, that is also 8 blocks. That is another kind of deep learning model. And yeah, so here if you see, they will predict the protein folding structure. So the blue parts they consider as high confidence score, and the green part they majorly look into the low confidence part. So this is the this is the 3D structure, and the confidence score, in the sense, this the predicting part, the blue is it, they are 90 to 95% sure that the fold look or the structure will be look like this kind of thing, the alpha helix will look like this. And the low confidence, in the sense, that part is they're 72, the confidence level is a little bit lesser here in this particular part. So yeah, so yeah, this is the basic that benchmarking and success in that incorporation of physical and biological principles here. They are incorporated in the particular AlphaFold architecture. So biological, in the sense, genetic information they are taking, as well as physical, like physics-based information, or the some coordinates and different angular momentum. Those things they are considering here. And then this is a end-to-end differentiable model. So this is a differentiable model, in the sense this is trainable. So then multiple iterative refinement stages they have done, although it is not completely out the model, but yeah, the overall Evo Permer architecture, those things are out. So from there we get these things. So yeah, these things, evolutionary sequence information, that multi-sequence analysis MSA part, basically they use the sequence and information from the input sequence. So this is how the AlphaFold architecture. So now we'll move into the just beyond drug discovery and protein synthesis. There are a lot of other things like genomics, genetics, and image analysis, biomedical signal processing, single cell genomics, metagenomics, then prediction of disease progression, personalized medicine, drug response prediction. So if we see into the machine learning community, each and every machine learning techniques can be utilized in different application areas of the biological systems, like the cancer means SBM kind of utilized in cancer, some classification techniques, then KNNs also multi-class tissue classification, gene expression kind of things. These are traditional machine learning techniques. It is not everything is deep learning. Only this part is the deep learning part: protein secondary structure prediction, amino acid sequence, or sequence similarity prediction, nucleotide sequence. So few of them means there are some data there where traditional machine learning techniques are also more useful. It is not like, if sometimes people think like the data is so huge, yeah, that research purposes, or the industry people or the pharmaceutical peoples, they have huge number of data. Why do we go for, as a student, as a researcher, then we have to look into whatever available resources we have, and based on that we can also look into different kind of machine learning techniques, and we can use them here. So if you see, the recently Google has a AI breakthrough that means that if you take a quick scan of your eye, that can detect the heart disease. That's alternatively going to the traditional CT scans or MRI and X-rays. So means that is more cost effective. So day by day, with a single mobile phone or single cell phone, we can able to do a lot of means I know one of the group Carnegie Mellon or something. They have actually, they are doing everything. All healthcare things should be done on the mobile devices. So you don't have to carry, if you want, if a doctor is going to a remote area to visit the patient, they don't have to carry huge instrument, or they cannot have those luxury of having those CT scan, all those things. But if they have a single mobile phone, they can use equivalent things of the preliminary stages of the diagnosis part. They could do it through mobile phone. So there are a lot of researches going on that way also. So yeah, then these are some other areas, those are quite means emerging, like genomic sequence analysis. Yeah, through this also people are trying to determine the disease, which gene is creating which disease. Then if they determine that gene, they could edit that gene so that that disease could be properly means we can cure that particular disease. So genomic sequence they basically analyze those sequences and try to determine which particular element or which protein is basically doing those things. So there are also in genomic, there are a lot of things like proteomics, transcriptomics, and there are a lot of things out there. So epigenomics, so there are a lot of things out there. Then precision medicine. Yeah, precision medicine, one of the hottest topic, and that is a very emerging and most hottest that research area in the healthcare sector right now. People are mostly looking forward to getting a medicine that is very specific to that particular person, rather than going a generic medicine, because generic medicine may not be that effective to the person. If we have the genetic information of the person, then we could determine, we can use those genetic information, and we could use that protein molecule for the particular disease. Then we could create a drug that is very specific to the particular person, rather than a group of person or a complete generic medicine. So that particular medicine will be given to the particular person, and it doesn't have any side effects because it is based on your own gene, and it doesn't also affect other persons, because the genetic information that the particular person is incorporated in developing that particular drug. So that thing, those information, those things can be done only through some AI based techniques, and those things now people are using a lot of that they're actually integrating those things, those data, and we have almost all data now. Human genome library is available, every gene sequence, everything has been already there. People know what, which gene is doing what function, or it is basically this particular gene is particular means how effective for this particular disease or something. So those things is done, and people are working on this particular precision medicine, and it is a quite interesting topic. Then this is traditional deep learning based medical image analysis and diagnosis. So yeah, a lot of we can capture a lot of images from X-ray to CT scan to MRI. There are a lot different kind of images we are getting. Then we are going through some deep learning, traditional deep learning techniques, computer vision based techniques, and yeah, you won't believe nowadays this particular models and architectures are giving much higher uh, performance than some experienced doctors, while they are actually once there was a challenge between experienced doctor and the architectural model, means AI based model. Then the AI based model gives 98% accuracy over the and the experienced doctor gives 95 or 96%. So they basically beat the experienced doctor. So yeah, that is not mean that it is replacing the doctor, it is mean to augment the doctor to give a much better diagnosis for the disease and all. So these are some ethical considerations we need to look while we go for any kind of biological research on this particular area, like data privacy, confidentiality. Those things are very essential. Then bias and fairness. There should not be any particular biasness of the data should be there, and data should be properly and fair data should be given, and transparency, responsibility, accountability. Yeah, and security. The security is one of the major concern here, because everybody's personal information is very means important, and as well as very it we if it means that the in security purposes, it is very essential to keep it in secured and not leaking those data and all. So those things, basically these are major ethical considerations and challenges we need to look. The confidentiality, also some legal issues, some like governance, confidentiality. So these are some legal part of these things, liability accuracy, decision making. Those things are the legal part also involved. So in the conclusion, future direction, so these are some, we need some interdisciplinary collaboration here, and this field is continuously growing. So, and the personalized medicine and the preventive healthcare ethics regulation, each part is itself is a, means ethics is also a kind of research area. AI for ethics, if you know that, that is research area, and that is most important in the healthcare sector. And definitely the global health. So yeah, this is the basically the interdisciplinary collaborations from the DNA to patient history to lab test, gene data, lifestyle, imaging, everything is interdisciplinary. So this is a huge multimodal. If we could create a multimodal system that incorporates all this data and give us a sophisticated diagnosis, and as well as basically I will say means diagnosing the patients and all, so those things will be much more effective. And yeah, these are some future advancements in these applications. One of the major thing is explainable AI. Till now, whatever we are doing means in normal traditional computer vision or NLP, we don't need the explanation that much, but in healthcare sector, the explainable AI carries a lot of weightage there, because we need to know why this particular gene is working that effective, or why this particular molecule is that effective on against this particular gene or something. That way, that is very essential, explainable AI. And these all are like integrated data analysis, advanced image signal techniques, and so these are basically. So that's from my side.
Thank you, Ramanarayan sir, for your wonderful talk. So, the participant, if you have any questions, so feel free to ask.
Hello.
Hello.
I just to know, like, if I have some knowledge on deep learning models and let's say some transformer models and some sequence to sequence models, and if I work in, let's say, explainable aspect of one of these data sets,
Yeah.
additional domain expertise do you need to assess, like, whether that explainability that I'm generating is meaningful or not? So, can you just give some clarity?
Yeah, I mean, see, to be honest, all deep learning models, they consider as black box. We don't know why this weight is happening, why this particular weight matrix is behaving like this, or why this particular, we don't know which feature actually giving much more importance. Suppose if you see in the healthcare, so we got a lot of genetic information, but we need to know which gene is most effective all genes. So if we go for this particular means deep learning models and all, they will feed all those genes and they will get the information or the features from the particular genes, and they use those features for determining or predicting that particular for the disease and all. But if we able to point out, okay, this particular gene or this particular protein molecule is more effective towards this, then it would be much more relevant for the people. And because there in healthcare system, there are a lot of redundancies there, lot of genes, a lot of data is there, but for at that particular time we need to explain it. Other than that, it is sometimes it is a ethical issue, like we don't know how it is happening, but we give it, but after sometimes there is some problem came up or some side effects came up of that particular drug, then it is not adaptable. So that's why I means you already need to know why this particular thing or this particular molecule, or why this particular feature is more effective for this particular
Yeah, that is in general. My question is actually like, let's say I'm working in text with some images. like we all understand, but when it comes to, let's say, protein folding or some drug related, some medical deficit,
Yeah.
so we may not have sufficient domain expertise to validate. Maybe we can have some evaluation metrics that we compute and then see. But my question is, like, let's say I want to enter into this this research area because it is very much related to all of us, the challenges and then the outcomes, and future directions look really interesting. So my question is, like, what domain expertise or the kind of thing that I need to study to start working with these kind of data sets to make some sense out of it, rather than just running a model and then reporting some results. That's what actually I mean.
Okay, means in healthcare sector or in biology sector, how you will approach?
Yeah, you are showing that big picture, like where so many challenges and other things that you are showing, right? Yeah, the very next slide. Yeah, maybe we can come to this slide. So
Yeah.
to play a role in any of these, like your beautiful idea, like when I'm in a remote place, I don't have access to a doctor, but still I can use one of these things and then have some preliminary responses and then some meditations that can be taken, right? So that's really a beautiful idea. So, like, to contribute in this kind of thing, what essential education do we need to start research in this kind of domains? That's the idea.
Okay, so first of all, I will say, as a ma'am, I'm just considering you as a computer scientist.
Yes.
Means you are a computer scientist. You don't have any.
And I have exposure to deep learning models. Let's say.
Yeah. Means AI scientist basically, and but you don't have exposure to this particular, I will say, biological systems and biological knowledge.
Yes.
Those things are there. So what happens, ma'am? If you look into the means initial phase, while you are targeting to the particular, means you need to pinpoint it, because whatever I am showing right now, it is very broad way I'm showing. Everything is itself is a research area, and everything is a very huge thing. So what happens, you need to pinpoint the particular research area you want to target, and then for that particular research area, like suppose you are thinking about genetic, basically gene analysis, genomic sequence analysis and all. So for genomic sequence analysis, yes, you need certain domain knowledge on those particular things. Other than that, you cannot understood why it is happening. You only, if you consider the data is itself is a data, I understand, you consider it is a data, the number of data points are there, the features are there, but you don't know the significance of the feature. So that significance of the feature only if you have certain, means it is not that difficult. Means if you go into some biological, because nowadays bioinformatics and all those things are coming up a lot, and a lot of video lectures and a lot of documents and a lot of papers are coming out of this particular bioinformatics. So basically, if you look into the some aspect, means for the particular means particular area you are targeting, only just you have to go through those particular means domain knowledge on those particular things, that is required. Other than that, you can also have a domain knowledge people in your team, and so that it would be much more effective and much more helpful to you, rather than yourself going into that particular. So that's how, means it's actually a very good question, means how to go into these particular things. And also it is very challenging, means every day you will get new problems, and those problems are unsolved problems. So you'll get a lot of exposure in this particular.
Okay, thank you.
So now, Ramanarayan sir, so we have one more question. So we have with us Professor Vapi Raju from IIIT Hyderabad. So he's the next speaker. So he would like to ask some question.
Okay, sure, sir.
Yeah, thank you, Dr. Ramanarayan. It's a very comprehensive lecture on possibilities of DL applications in this biological domains. Two questions, general questions I have. One is, are you aware of any pre-trained models. I mean, this is the age of availability of pre-trained models in, for example, language. We have large language models which are pre-trained available for people to tune for different applications. Are there any that you know of for in protein structure prediction or any of these domains? Are there any large pre-trained models available for researchers? Second question is, yeah, maybe I'll just give the second also, then you can.
Okay.
The second question is, in general, one of the important areas I feel is the multimodal representation learning.
Correct.
Especially, the interface between health and biology.
Yes.
For example, if you take diabetes or something like this, there is a genomic counterpart, there is blood biomarker, then there is lifestyle questionnaire. And what is the state of the art in combining these and learning multimodal representation for risk prediction or any of these? Right? Thank you.
Yeah, thank you, sir. So coming to the first question, yes, there are a lot of basically lot of pre-trained models are available. So, but it depends on which, basically, as you asked, protein folding models, protein folding. Yeah, you can get pre-trained data for AlphaFold 2. Then you can go for any kind of other than pre-trained model. There are some open source models are also available, like OpenFold. OpenFold is a open source models. AlphaFold is not open sourced. It is, they have only given the weight matrices and the pre-trained data. But OpenFold has some open source models. There you could tweak and you could use it for your own purposes, and you could use for your own data. So those things are available nowadays. Means, yeah, but it is not that expensive as it is like LLMs and computer vision models, because this is actually in nascent stage, and people are actually working a lot in this particular domain. If you see, the research is means even if you see into the Google, Microsoft, Amazon, everywhere they have a science domain team. So they are majorly working in biological sciences, biological, chemistry, physics. So those things are there. And coming to the am I able to answer your question?
Yeah, whether my question is how much of that is accessible to academic researchers like me, if they are, available, how much of it is available? Like, for example, your answer OpenFold has model is also open. That's very important to know. Any other domains where there are
Yeah, means yes, you can actually for individual, means the genetic this particular I will say protein folding structure rather than protein folding. If you go for the RNA sequence analysis and all, so they are also based on individual task or the individual objective. You will find a lot of, means, because some are some big models and some established models. Those pre-trained models also available, and there are a lot of open source community are being established here. So there is a team called M2D2, molecular modeling and drug discovery. That is a consortium is there. In that consortium, if you see, majority of the company industries and some top-notch academia like Mila, Stanford, some big groups like Broad Institute, MIT. So those are actually creating consortium, and they are actually doing the research, and they are publishing the things. They are, means, publishing it in different conferences and all. They are putting it all the data, everything there. So there are a lot of things are coming up like this for specific applications. If you look into the individual specific application, you will get. And if you look into the generative AI part, there are a lot of work is happening in this generative AI part also. You will get a lot of very good models along with the code, means open source code also available, lot of cases.
Right. Thank you.
And in the second question, yes, the multimodal data that you asked, that is actually quite interesting part. But till now, every unimodal data part is not completely solved. So yeah, the multimodal part, if you look into the EHR, electronic health record, that electronic health record basically dealing with multimodal data, that basically dealing with the images basically taken through any kind of CT scan, X-ray and all. Also the prescription, the drugs you have taken, they are also trying to incorporate the genetic information there, so that it could give us a complete multimodal. And people are working a lot on this particular EHR domain. But yeah, I haven't seen that huge thing has come up, just because till now in traditional manner, if you see the multimodal thing, just now Google just released the Gemini. So it is a long way to go into the biological healthcare sector, because till now the normal NLP, computer vision, those things has not been done that way. So once, but it is on the way, basically, I will say. There are a lot of research is going on, and it, in near future we'll get some kind of.
Right. Thank you very much. Thank you. Thanks.
Thank you, sir.
So thank you very much, Ramanarayan sir, for your wonderful talk. So, your talk was really informative. So I hope that in upcoming such events, we would again invite you. So thank you once again.
Thank you, sir, for giving me this opportunity. This is really mean a lot to me. I really enjoyed this particular
Yes.
discussion and all.